Hey guys, so let's see if we can delve into the world of chi-square a little bit and maybe offer some clarification, and hopefully everybody can now be on the same page. So I'm going to run through some notes. I'm going to go over the formula. I'm going to define what chi-square is. We will do a couple of I throw one big genetics problem up there. We'll talk about all the parts and pieces, like degrees of freedom, critical value, all of that. We'll talk about what you do with that final number, how to reject or accept your null hypothesis, and then we'll work some practice problems at the end. Okay, so the first thing you have to realize is that chi-square is a statistical tool. And that tool is designed to help us decide if the observed values that we see when we do an experiment or when we make a count or whatever is close enough to the expected values to be acceptable within a scenario. You can calculate or you can utilize chi-square analysis in lots of areas of biology. You see it in genetics, but it's not just a genetic tool. Any place that you are comparing expected ratios and observed ratios where you're looking at the difference between sets of numbers, you can utilize chi-square. You guys saw this on your mock test when they made you apply chi-square to the behavior of fruit flies. Did they prefer to go to one chamber versus another chamber? So as long as you have at least two variables, you're comparing at least two things, and you're generating numbers from that, and you can talk about this is what I thought I would see versus this is what I actually saw and counted myself, you can utilize the chi-square analysis for anything that falls into that category. So on the screen right now just shows the formula that we use and a breakdown of what all the parts and pieces are. And then it shows a graph comparing probability to chi-square and the levels of significance of where these values could fall. So how statistically different something is versus how statistically not different something is. You will see this formula again in a little bit. So if you want to write it down and write out the parts and pieces, it is coming back in just a little bit. So one way to think of chi-square is to think that it allows you to figure out if the data that you collected is good or non-biased, or if that same data is quote-unquote bad or biased. So if statistics show that your data is biased or it is bad, it just means that somehow the data that you collected, the numbers you actually counted yourself, is very different from what you would have expected based on that scenario. Okay? And pretty much you're trying to figure out if something is causing that difference beyond just, oh, normal luck or normal chance, normal occurrences. So is there something else behind the scenes causing the numbers that you are seeing? So if we think of flipping a coin or maybe rolling some dice. For flipping a coin, we're dealing with two sides. It's either going to be heads or tails if it's a normal coin. So if I flipped a coin 100 times, I would expect, without actually counting, just based on the fact that it's two-sided and I'm flipping it 100 times, I would expect that 50 of those times my coin would land on the head side and 50 of those times my coin would land on the tail side. That is my expectation. If I went ahead and actually flipped my coin 100 times and counted how many times I landed on heads versus how many times I landed on tails, and there is a very big difference between my expected value of 50 and what I actually do, it suggests that something more is happening with this coin flip than just it happened to land on heads or it happened to land on tails. It could be that something's wrong with the physical coin itself, or it could be that something is wrong with the method in which I am flipping it. I am purposely doing something to cause it to land in a specific way. Now, when we did our corn lab, where you looked at that yellow corn and the weird looking purple corn, and some of them were wrinkled and some of them were smooth. So hopefully you remember what I'm talking about. We used the laws of probability to determine all of the different outcomes based on what we knew the parents of those corn plants to be. So if we were to take our corn data, how could we figure out if our data was good? Because you physically counted a certain number of rows and you counted for different phenotypes. Some of you were looking at color, some of you were looking at the shape of the seed, some of you were looking at both, but you literally went down row by row and counted how many seeds were purple and smooth, how many seeds were purple and wrinkled, how many seeds were yellow and smooth, how many seeds were yellow and wrinkled. And you actually had numbers to represent those four categories. But you also had a hypothesis, a thought, let's say, about what kind of genetics the parents that produced this corn plant would have had to give you the variables that you are seeing, or to give you the phenotype that you are seeing. So how do you know that what you counted actually matches up with what you expected? Or how does it match up? Is it a good fit? Are they close together? Or is it bias? Are they far apart? Are they not a good fit? Well, the only way you can truly tell that is by running or performing a chi-square analysis. All right, so here's the formula for chi-square. And chi-square is a summation, so the symbol that we use to represent chi is actually the Greek letter x. Well, not the Greek letter, it's the Greek letter chi, the letter we know as x, and we square it. So x squared, this right here, is our symbol for chi-square. Hold on a second, let me change pens. Okay. Now, this is a summation, which means that once we get a certain set of values, we will start adding all of those values together. So our little weird looking E is the summation sign. It just means that we are taking a sum of all of our numbers. Now, if we look at this section of the formula, it has, we can say, two variables in it. It says O minus E, all squared, divided by E. Your O represents your observed individuals with a given phenotype. Now we're saying phenotype because the examples that we're using are genetics problems, but remember, it doesn't have to be genetics based. So it could be the observed individuals that performed a certain task or did a certain behavior, or exhibited a certain behavior, like moving over to the wet side versus moving over to the dry side. Then your variable E represents the expected number of individuals with either a given phenotype or who would have performed that certain activity or that certain mode of behavior. All right, so E here and E here represent the same thing. It's still going to be expected individuals with a given phenotype or a given behavior. All right, so if you need to real quick, go ahead and write your formula down and write out what all the parts and pieces are. Okay, so another part of the puzzle with the chi-square situation is going to be what we call the null hypothesis. So the hypothesis in this case is called the null hypothesis, and it pretty much states that you should not see any real difference statistically between your observed values and your expected values. It should be an even playing field. So we go back to that example where I said I have a coin that's two-sided, I flip it 100 times. I would expect that I would have an equal number of lands on heads versus an equal number of lands on tails. My coin should land on heads at the same amount of times that it lands on tails, based on the fact that I flipped it 100 times and it is two-sided. So I'm expecting 50 tails, 50 heads. That would be an example of a null hypothesis. There's no real difference between what I observe and what I expect. Okay, that's my expectation. I'm going in thinking that when I actually do it, I will see numbers very close to 50 for both heads and tails. That would be my null hypothesis. Nothing's different. Okay, everything should be about the same. So in other words, the null hypothesis just means that your results or any differences that you do see between what you observe, so the numbers you've actually counted, and what you went in expecting are just caused by randomness. It's by chance and only chance. Lady luck, if you will. There's nothing else causing those differences. I'm not flipping my coin a certain way. My coin really is two-sided. Like the earth didn't change axes. Nothing weird has happened. It's just completely by chance. That's what the null hypothesis states. So once you work out all of the different parts of chi-square in terms of the actual math, and you end up with a chi-square value, you really only have two outcomes. Either your null hypothesis is supported by your data set, or it is not supported by your data set. So after you've run your chi-square analysis, either it's in support of your null, or it is not in support of your null. So if your null hypothesis is supported by your chi-square analysis, you are just making the assumption that what you're seeing is because of randomness. So if we're talking about corn or fruit flies or people or animals or whatever, we're going to assume that the reason we see the phenotype that we see is because the mating process is random and genes segregate and they assort independently, which just means that if you think back to meiosis, where we talked about genes, when we talked about chromosomes, moving apart in like anaphase, for example, that is a random process. You don't know what part of the chromosome is going to end up where. When we talk about they independently assort themselves, you don't know which of the chromosomes is going to end up in which daughter cell, which will eventually produce an offspring. So those two processes are just due to chance. It's just due to randomness. If you think of your family, if you belong to a family that has multiple people in it, so mom and dad and a couple of brothers and sisters, if mom and dad are the parents, biological parents of all the siblings, it still doesn't guarantee that all of them will look alike. Think of your brothers and sisters. You probably don't look exactly alike. And it's because your parents have different alleles, different versions of the same gene. And they really don't control which version which child gets. Your mom doesn't sit there and think, okay, so my first daughter, I want her to have blonde hair, so let me go ahead and send that blonde hair gene. And then I want her to have blue eyes, so let me go ahead and send that blue eye gene, and I'm going to make sure it's going to get into this very first egg that's going to get fertilized. That's not how the process works. You end up with what you end up with because it's completely random. So if your null hypothesis is supported by chi-square analysis, this is pretty much what you're expecting, okay? You're saying that everything I see is because mating is random and because chromosomes behave in a random way. They segregate, they move apart from each other randomly, and they go into different daughter cells randomly. It's independent, no control by the parents, okay? Realize that when we're talking about genetics and biology and we're utilizing chi-square, this is always going to be our assumption. We are always going to assume that in a genetics cross, mating is going to be random, genes are going to get segregated, and they get moved around or assorted independently. Okay? This is how normal meiosis works. Now, if your null hypothesis is not supported by your chi-square analysis, it means that the difference between what was observed, so the numbers you counted, and what was expected, the numbers you went in thinking that you would see, are really far apart. There is a very big difference. That suggests that something non-random must be occurring. The difference in number is caused by something more than just your parents mating with whoever they got attracted to and the genes and chromosomes moving around independently. So some possible explanations for what that something else could be that some of your genes are not randomly segregating because they're sex linked. Remember, some things are found on that X chromosome, and once they're on the X chromosome, wherever the X goes, they go as well. They are linked to that particular chromosome. Some genes are linked together, even though they're not one big package, but they behave like they're one big package. So if one part of the gene gets inherited, it means the entire thing gets inherited. So instead of inheriting one trait, you actually inherited 3 different traits. The other thing that we can think of that can cause some non-randomness would be mutations. Okay, so all of these could be possible explanations for why you're seeing that big varied difference between your numbers. So once you've gone through and you've calculated a chi-square value, you're then going to want to compare your chi-square value to something. That's how we determine if the analysis is going to support your null hypothesis or if it's not going to support your null hypothesis. So instead of having you commit a bunch of numbers to memory, everything is placed in what we call a probability chart or a chi-square chart for you to use. But it's not necessarily clear-cut and simple. In order to correctly use and determine, or in order to correctly determine the probability, you have to be able to calculate what is called the degrees of freedom value. So your degrees of freedom is abbreviated DF, and it's the number of phenotypic possibilities in your genetics cross minus 1. Now this is assuming we are applying the chi-square technique to a genetic situation. If we are not applying it to a genetic situation, like again on that mock test, when you're looking at the distribution of your fruit flies or roly polies or whatever, then it's going to be the number of different possibilities that you could have in your scenario minus 1. Okay, so hopefully that makes sense. So on that chi-square chart, one of those columns or rows, just depending on how the chart is organized, should say degrees of freedom or DF. And once you've done your little subtraction, your total number of possibilities minus the number 1, then you will find that DF value. So let's say it's 1 or 2 or 3 or whatever. You'll just, on that table, you will find that DF value. And then you need to use the probability or distribution to figure out what we call your critical value. So in science, normally the level of significance that we like to read from is 0.05. So on that chi-square chart, under probability, you are going to look for the column or the row, again, it depends on how the table is oriented, that is headed up by 0.05. So if your level of significance is greater than 0.05, then the null hypothesis is going to be accepted, and we're pretty much saying that the results we see are due to chance alone, and they are unbiased. But if the level of significance is smaller than 0.05, then the null hypothesis is going to be rejected, and the results are due to something else, random chance. Alright, so let's look at an example. So this is an example showing a dihybrid cross between fruit flies. And we are crossing a wild-type fly that has normal eyes and normal body color with a fly that has a black body and is eyeless. So no eyes, and instead of being like an orangish yellowish color, the body is like a dark brown to a black color. So these flies right here would be our parentals. This cross would produce an F1 generation that all has the phenotype of wild type, meaning all normal, red eyes, normal body, but they would still carry the genes for this particular, let's not necessarily call it a mutation, but for this particular phenotype of black body with no eyes. So if we were to cross the F1 generation with each other, so cross 2 heterozygotes for other words, this would be the result. We would get some flies that are wild type. We would get some flies that are eyeless with black bodies. We will get some flies that have normal bodies but are eyeless, and some flies that have normal eyes but have a black body. So notice the numbers. We have 5610 flies that are wild type, 1881 flies that are normal and eyeless, 1896 flies that are black-bodied and eyeless, and 622 flies that are black-bodied with normal eyes. All right, so in this one, it's almost like you're working backwards a little bit. You go in first and collect your data. So you've already collected your observed numbers. Those are the numbers we just looked at. Once you know the total number of offspring in each class, once you've counted it all, you need to determine your expected value for this particular cross. So you would actually do the dihybrid cross, and you would realize that you should have a 9 to 3 to 3 to 1 ratio. 9/16 of your total population should be a wild type fly. 3/16 of your total population should be normal bodies with no eyes. 3/16 should be black bodies with normal eyes or wild type eyes. And 1/16 should be black bodies with no eyes. That's your breakdown in terms of your ratio. Okay, so you can use these values, these sixteenths, if you will, so 9/16, 3/16, 3/16, and 1/16, to figure out what your expected numbers would have been if you had not already done this experiment. What would you have gone in expecting under these conditions? All right, so now we actually have to conduct the analysis. So here we have all of the different phenotypes that we are looking at. And here we've already inputted all of the numbers that go with those phenotypes. And then this number was our total. Now, another way to think of hypothesis would be as your expected value. This would be your e-value. What was I anticipating this population would look like, not knowing these actual numbers? So you would take your total population number, that 10,009 number, and you would multiply it by the excuse me, the ratios that we had come up with of 9 to 3 to 3 to 1, or 9/16 to 3/16 to 3/16 to 1/16. So it would be 10,009 times 9/16, 10,009 times 3/16, times 1/16. And that would give you your hypothesis or your expected values. So this slide just goes over pretty much what I just talked about on the last one. This is how you would calculate your expected value. So again, your total fly population is 10,009. To figure out your wild type population, because we've run the Punnett score, so we know that 9/16 of our cross should be wild type. It would be 10,009 times 9/16, which would give you 5,634 flies. Then your eyeless flies would be 1,878 flies. Your black body flies would be 1,878 flies. And your black body and eyeless flies would be 626 flies. So these four numbers would represent your expected values. This is what you would have thought your results would have looked like, assuming that there was no difference, there was no discrepancy. All right, so now you can compare through this table exactly where our observed numbers, or how our observed numbers, compare with our expected numbers. Okay, so O numbers versus E numbers. And just by a quick glance, it doesn't look like it's too much of a difference. So our null hypothesis should say that the two traits that we are testing, black body and eyelessness, are not actually linked, and we think that they would have randomly segregated and randomly assorted during the formation of this gamete, during the formation of this new cell. So the differences between our expected values and our observed values are due to chance alone. We don't think that there's anything more going on than just normal meiosis. That's what this null hypothesis states. Okay, so now if we went through and did the actual calculation for chi-square, remember the formula says that it's a summation, first of all, but it's the observed numbers minus the expected numbers, all squared, divided by the expected number. And for each different phenotype, the resulting number of this part of your formula, we're going to add all those numbers together to give us chi-square. So in the first part, it would be, well, it's laid out for you. I'm not going to read it out for you, but here it is. It's all laid out for you. This would be the math behind, whoops, sorry. So this would be the math behind the numbers that I just read off and, we just looked at. And then when we add them all together, our chi-square value is going to be 0.3 or 0.3. So now the next step would be to calculate how many degrees of freedom. Because we can't figure out our critical value, we cannot use that chi-square table until we know how many degrees of freedom. So there are 4 different phenotypes, and we know that it's the number of variables minus 1, so the number of possibilities based on our scenario minus 1. So that would be 4 minus 1 to give us 3 degrees of freedom for this particular example. Okay, so now what? So if you remember, I told you that the part of the table, the probability, that we care about is this 0.05 column, which is right here. Okay, so that 0.05 is probability, it's our p-value. And we also have to look at our degrees of freedom, which in this case would be 3. I think I did a pretty good job with those lines. Y'all should be proud of me. Anyway, so where these two lines meet or intersect, right here, that 7.82 represents our critical value. So now we are going to compare our chi-square value to that 7.82 value. We're going to look to see, is it greater than or lesser than? Is the chi-square value greater than or lesser than? So to remind you, our chi-square value that we just worked out was 0.3. Okay? So 0.3 is definitely smaller or lesser than 7.82. We'd have to go, let me switch colors real quick. We would have to go in this direction to run into 0.3. It's actually even smaller than over here. So we would need to accept this null hypothesis. The difference that we saw in those values were so minuscule, so small, so close together that we are going to go ahead and say that any differences, any deviation is due to just random chance. Okay, there is nothing more going on than just normal meiosis. Now, if that value had been greater than 7.82, so let's say it had been 9 or it had been 15, then we would have gone in the opposite direction where we would reject our null hypothesis. And if we did that, we would be saying that no, the differences we see between our observed numbers and our expected numbers are so large that it can't be normal meiosis. There is something else going on. There is a mutation of some sort. There is some kind of linkage between the genes. Something more is causing this difference in numbers. Okay, so this is just the word version, like the written down version of what I just said. So if we look up our statistic, our chi-square value that we came up with of 0.3, we're looking at it on that chi-square distribution table. This is the following information that we would get. The p-value is 0.05. Based on the scenario, there are 3 degrees of freedom. So using that table and using a combination of those two values, we can therefore figure out that our critical value is 7.82. So now we have to compare our chi-square value to the critical value. If it's smaller, it means that about 95% of the time, when our observed data is this close to our expected data, the difference we're seeing, that deviation, is because of randomness. It's just chance, it's just normal mitosis, there's nothing else going on. So because our chi-square value was so much smaller than our critical value, we are going to go ahead and accept our null hypothesis. By accepting it, we are saying that there really isn't any big difference between the two sets of numbers that we worked with, between our observed numbers and our expected numbers. The little bits of deviation are caused by just random chance, just normal mitosis. All right, so we've talked about degrees of freedom, we've talked about null hypothesis, we talked a little bit about probability, but we haven't really talked about what this critical value thing is. So the critical value is pretty much the point at which we would reject the null hypothesis. How high are we willing to go before we say enough is enough, there's something more going on here? And it is always based on your probability that you work with. So in the case of us, we're going to be working with 0.05, and how many degrees of freedom you have in that problem or in that scenario. Now, your degrees of freedom will change all the time. It just depends on how many possibilities you have. So if we go back to that example that I was giving before, where I was talking about flipping a coin, then there are only two possible outcomes. Either I land on heads or I land on tails. So in a scenario like that, my degrees of freedom would be 1, because it would be 2 minus 1 equals 1. If I had changed and I was working with dice instead, let's say I was casting one die. Well, one die has 6 sides. So I could have 6 possible outcomes, 1 through 6. So now my degrees of freedom would become 5, because it's 6 minus 1 equals 5. So the degrees of freedom just changes depending on the scenario that you are in. Now, if our chi-square value was 0.0, what am I saying? If our chi-square value was 8, but we had 4 degrees of freedom, would we accept or reject the null hypothesis? Well, if we went back to that table, we would see that we would still accept the null hypothesis, because if we change our degrees of freedom, we would change the critical value that we have. So our new critical value would be 9.48, and a value of 8 would still be smaller than 9.48, so we would still accept that what we're seeing is due to chance. Okay, so how do you write all of this up? There is a very particular way I would like you to report this data, and it's using the stuff that's like written in red. So for every chi-square that you solve, this is what I'd like you to say. With blank degrees of freedom, so that's where you would insert how many degrees of freedom you have based on your question. My chi-square value is blank. So this second blank is where you would tell me what your chi-square value is. Then you would go on to say which is greater or lesser than the critical value. So you would choose one depending on what it is. If it's greater than, if it's bigger than, you'll say greater than. If it's smaller than, you'll say lesser than. Okay, then I would like you to state what your critical value is. So 7.8, 3.6, 12.79, whatever. Therefore, I would either accept or reject my null hypothesis, and you choose accept or reject based on whether it is bigger than your critical value or smaller than your critical value. So again, you need to always state how many degrees of freedom. You need to state what your chi-square value is. You need to determine if your chi-square value is bigger or smaller than your critical value. You need to state your critical value, and then you need to choose whether you would accept or reject your null hypothesis. Okay? Then at some point you would also want to explain the significance of the data. So that's where you would go back to whether or not the deviation or the difference seen is caused by random chance or something else like a mutation or linkage or whatever. All right, so I'm done with the notes section for now. I hope this makes it clearer. By all means, if you need to come see me next week, please do so. I'll be there. Come find me. So now we're going to run through some problems and see if we can get a better understanding for chi-square. So I'm going to... All right, so let's look at some genetics problems. We're going to look at peas. Hopefully I can write on this thing without embarrassing myself. So we have yellow peas. Okay, and yellow peas are going to be dominant. And we have green peas. And we're going to cross two heterozygous plants. So if we cross two heterozygous plants, we're going to have mom and dad being big A, little a. Okay, so this would be the outcome of that cross. Now we're only looking at phenotypes, so we only care how many or what the ratio of yellow plants or yellow peas are, to green peas. So our yellow peas, let's see, we will have 3 of them. So 3 out of 4. And our green would be 1 out of 4. So our ratio is 3 to 1, or 3/4 and 1/4. Right, so I'll give you a minute to copy that down if you're still working. Now, we've done this experiment. We have crossed these 2 plants, and we have some numbers. We have 4,400 yellow peas, and we have 1,624 green peas. So if we were to run chi-square between our observed numbers and our expected numbers, what would be the result? So let's do it. So these numbers over here, I should have made this smaller, but this represents your observed numbers. And let's go ahead and put our expected numbers, hope you can see that, right there. So how would we figure that out? Well, we first need to know the total of what we're working with, and that works out to be 6,024. Earlier, we figured out the cross, and we know that we had 3/4 of our plant, or of our peas, I keep saying plant, sorry, 3/4 of our peas being yellow, and we had 1/4 being green. So we would just have to take this 624 number and multiply it by 3/4, or by 0.75, or by 1/4 to figure out our expected values. Okay, so if we did that, we should get 4,518 yellow peas and 1,506 green peas. So if we ran chi-square, we would do 4,400 minus 4,518 all squared divided by 4,518. And then we would do 1624 minus 1506 all squared divided by 1506. And the result that we would get for this upper section of our table would be 3.08 and then 9.24. Okay, which would give us a chi-square value of 12.32. Hold on one second, guys. All right, so before we even go to our table, let's figure out our degrees of freedom. So we know that the degrees of freedom are equal to the total possible outcomes minus 1. So in this scenario, you could only be 2 things. Either you were a yellow pea or you were a green pea. So our total possibilities would be 2 minus 1 to give us 1. So we have 1 degree of freedom. We know that our probability, our p-value, is going to be 0.05. So now we just need our chart to be able to figure out what critical value we're working with. Okay, so here is our chi-square table. We know that we are paying attention to this column right here. That's our column of interest. And in this particular scenario, we only had 1 degree of freedom, so it means that this is the section that's most important to us. So our critical value is going to be that number right there, 3.84. Okay, so let's see. CV, critical value, is going to be equal to 3.84. We came up with a chi-squared value of 12.32. So we are comparing to see if this chi-square value is higher or bigger or greater than our critical value of 3.84. And yes, it is. It is bigger. So it means that we would be moving in this section of our table, right? Which makes our discovery significant. According to this table, it divides it into significant and non-significant. That's not really a big deal. Excuse me, sorry guys. I ended up yawning. But for us, we are going to have to do something in terms of accepting or rejecting this null hypothesis. Well, if we've been accepting the null hypothesis when it's been over here, if we are now on the opposite side, it means we are going to reject this null hypothesis. We are rejecting what we are seeing because we feel, based on our analysis, that these results are not random. They are not caused by chance. They are caused by something more. Okay, it does not fit in with our predicted data. All right, so I hope that makes sense. Okay, let's look at another example. So in this one, we're gonna work with some corn seeds. We have 4 different phenotypes. So in this corn plant, purple seeds are dominant to yellow seeds, and smooth seeds are dominant to shrunken or wrinkled seeds. So we're going to end up with seeds that, we're going to end up with seeds that are purple and smooth. Really bad handwriting tonight, guys. We're going to end up with seeds that are purple and wrinkled. We're going to have seeds that are yellow and smooth. And we're going to have seeds that are yellow and wrinkled. Okay? When we ran the cross and we counted the result, we ended up with 216 purple and smooth seeds, 79 purple and wrinkled seeds, 65 yellow and smooth seeds, and 21 yellow and wrinkled seeds. Okay? So, run a chi-square figuring out if we were crossing, or how about this? If we were crossing 2 heterozyguses, let's say that our ratio is going to be 9 to 3 to 3 to 1. Because sometimes you're given the ratio. Sometimes you don't have to worry about all of that. So, it's going to be the same step as before. Actually, I'm going to erase this and write it someplace else. Because that way I don't have to set the problem back up. So we have a 9 to 3 to 3 to 1 ratio. Okay, so these are our observed numbers here. And let's put our expected numbers there. And the way we would do that, we'd have to start off by figuring out what is the total number of seeds that we are looking at. So we're looking at 381 seeds total. So we're going to use this total, and we're going to do some multiplication utilizing our ratio for us to get our expected numbers. So when we do 381 times 9/16, we get, oh, maybe I could do this in a different color to help you out, but we get 213 seeds. When we do 381 times 3/16, we get 72 seeds, so we'll have 72 twice. And then when we do 381 times 1/16, we get 23 seeds. Okay, so if we went through and ran our chi-square, O minus E, all squared, divided by O, the result should be 0.04, 0.68, 0.68, and 0.17 to give us a chi-square value of 1.57. Okay, so we now have to calculate our degrees of freedom. And degrees of freedom, as we know, is equal to the total number of possible outcomes minus 1. In this case, there are 4 possible outcomes, so 4 minus 1 equals 3. We have 3 degrees of freedom. So now we know our probability is going to be 0.05, because it's always 0.05. So let's go to our chi-square table and figure out what we should do with our null hypothesis. Alright, so here is our chi-square table. Here is our probability column of interest, okay, because that's the 0.05. We are looking at 3 degrees of freedom, so right there, which means that our critical value is going to be 7.82. So the critical value is 7.82. Our chi-square value was 1.57. 1.57 is smaller than 7.82, so it means we are moving that way, which means that we would accept our null hypothesis. Okay, and that's just saying that any differences we see between our observed numbers and our expected numbers is due to random chance, nothing more. Alright, so I hope this was helpful. Feel free to view this as many times as you need to. Feel free to come see me next week with any problems. Just whatever it takes to get this to a point where, or a place where you guys feel comfortable. Alright, so hope to see you next week. Bye.